In this module, you will be learning about biosecurity and biosafety. Some incidents of biosecurity will also be discussed. People who work in close proximity of infectious agents, as in hospitals or research laboratories, are required to take courses in biosecurity to protect their own health by, first, preventing their exposure to human pathogens, second, preventing the release of microorganisms into the environment. Therefore, biosecurity could be defined as measures taken to reduce the risk of infectious agent theft and dispersal by laboratory staff or others. On the other hand, biosafety means the protection of laboratory staff from being infected by pathogenic microorganisms. The common understanding of biosafety is derived from the practical guidance issued by the World Health Organization on techniques for use in laboratories. The World Health Organization Laboratory Biosafety Manual considers biosafety to be the containment principles, technologies, and practices that are implemented to prevent unintentional exposure to pathogens and toxins or their accidental release. We've already learned historical cases of using microbes as a bioweapon. In this topic, you are going to learn about a most recent incident of biosecurity where a person with high profile could actually, out of revenge, infect someone with AIDS. Let's take the case of Louisiana versus Schmidt as a biosecurity incident. In this case, HIV-infected blood was used as the weapon in an attempted murder. This case is instructive for the forensic microbiology system. A vial of HIV-infected blood was found in the office of a suspect, a gastroenterologist. The challenge for forensic microbiologists was to provide evidence that this was or was not the source of the victim's HIV infection. HIV is an RNA virus which undergoes rapid mutation, so any direct genetic comparison of the donor source and the victim is complicated. In this case, analysis focusing on both rapidly and more slowly mutating gene of HIV proved to be very useful. Examination of strains from the vial, the victim, and control samples revealed that the viral RNA from the victim was more closely aligned to that from the vial in the suspect's office than to viruses from other patients in the area. The clinical history and clinical laboratory data obtained by attending physicians provided supporting evidence of the uninfected status of the victim prior to this injection. But how? The victim's prior HIV-negative status was documented by blood donation screenings and negative HIV tests of sexual partners prior to the injection. The evidence was presented in a U.S. criminal court. Based on the composite epidemiologic and forensic microbiology evidence presented, a conviction for attempted murder was obtained. Remember, in any cases involving a bioweapon, both the person who collects the evidence and the later analysis is very important for solving crimes. Sample collection, documentation, and preservation are vital to the biocrime investigation. Microbial evidence can be informative, but it's rarely the sole deciding evidence. When considered in conjunction with other evidence, in this situation, epidemiological and clinical data, the case was very strong. Instructive lessons can also be found by reviewing other cases in the literature, such as the laboratory technician who poisoned her co-workers with a laboratory stock of Shigella dysentery type 2 in muffins, and the poisoning of salad bars to skew an election for political gain. This will be studied in the coming topics, so hang on, more interesting cases are to come. In regards to biocrime, educating the public with the basic knowledge of microbes and the infectious diseases we face in our daily life is important to deal with any intentional spread of microbes. In 2003, a study was done by Blinden and colleagues to evaluate knowledge and beliefs about smallpox virus, vaccine against the virus, and possible reactions to an attack in the USA. They've done a survey by randomly selecting 1,006 adults. The results of the survey were alarming, as the authors concluded that a majority of respondents had misunderstanding or wrong beliefs about smallpox virus and vaccines. In the next topics, you will learn some other biosecurity incidents to understand the magnitude of the matter and to appreciate the importance of educating the public regarding microbes. To conclude this topic, in biosafety, the focus is on protecting humans. However, in biosecurity, the focus is in preventing the intentional release of microbes, thus securing the microbes in laboratories. Biosecurity incidents are important to study to understand the concept.